Good morning. My name is Yu from UCSD Bioengineering. I'll be here talking about our work that has the potential to revolutionize the way we perform solid organ transplantation and to address the challenges of allograft rejection. So, as we know that solid organ transplantation has been a beacon of hope for patients with end-stage organ failure, dramatically improving patient survival and quality of life. So immunosuppressive regimens, while effective in reducing the incidence of rejection, associated with a range of potential life-threatening side effects, including cardiotoxicity, nephrotoxicity, and diabetes. So a paradox arises in which the solution to one problem can lead to another set of equally serious challenges. So the biological question is, how can these risks be reduced while improving the organ transplanted outcomes? So the focus of our work turns into an emerging strategy that offers a promising answer. So the infusion of regulatory T cells, Tregs, alongside SOT. So this approach aims to rebalance the Treg ratio, modulate antigen-presenting cells, and promote tolerance within the transplanted organ. So thereby preventing rejections or loss. However, a major obstacle arises lies in our limited understanding of the in vivo biodistribution and persistent efficacy of these infused cells. So traditionally, the visualization of cellular subpopulation in vivo has relied heavily on proton MRI. So particularly paramagnetic metal ions like ferromoxidol, as we see here in the lower side of the right figure. So they have their limitation, including the need of extensive manipulation of cell culture and relatively low sensitivity. So there come a great gap to solve. We use the CD25 microbead that's composed of CD25 antibody and texture-coated ion beads with average diameter of 50 nanometers. So what is truly remarkable about these beads is that they can be internalized by Treg cells, resulting in a strong T2 signaling change that can be detected on MRI images as shown in figure B. So our initial results are promising that the Treg cells labeled with CD25 microbeads had attenuated signals on T2-weighted MRI compared with unlabeled cells and agarose control. So which is consistent with labeling with paramagnetic ion oxide. So the corresponding T2 relaxation maps showed quantitative T2 signal attenuation. 500,000 Tregs had a significantly lower T2 signal than those of unlabeled T cells and agarose alone. So the T2 signal of the million labeled Treg sample was further shortened, as shown that we have demonstrated that these microbeads can be effectively used as MRI contrast agents, providing improved sensitivity and specificity for imaging cellular subpopulation in vitro. Now we jump to the in vivo part. The mice receiving CD25 microbead labeled Tregs showed attenuated liver signals on T2-weighted MRI images 24 hours after injection, after the systematic injection, compared with negative control mice receiving the CD25 bead labeled T cells. So the mice injected with CD25 beads alone also showed a strong T2 signal attenuation 24 hours after injection. So quantitative analysis of T2 values in the Tregs mice receiving the CD25 beads showed the significant changes in T2 values between pre and 24 hours after MRI. So this change was not observed in CD25 labeled T cells. So our stats analysis comparing T2 changes in all mice has confirmed the significant signaling change in mice receiving Treg compared to T cells and the microbeads alone. So these results confirm the feasibility of tracking Treg in vivo. So to conclude that our finding of the one-stop shop approach utilized the MR properties of select beads to precisely track the movement and the distribution of Treg cells across a wide range of disease states. So imagine the possibility from a clinical imaging perspective, we are able to understand the mechanisms of inflammation and immune response with unprecedented clarity and improve our ability to diagnose and treat complex disease with Tregs. Thank you. I want to thank my, this is my lab.